Hello, and welcome to this episode of our Active Motif webinar series. My name is Mason Brooks, and today we'll be hearing from my colleague, Dr. Joshua Messenger, who is an R&D scientist here at Active Motif. Today he'll be talking about strategies for enabling epigenetic data to be used not just in research, but in the clinic as well.
Hi, Mason. Thank you so much for that wonderful introduction. I'm excited to be here today to talk about a high throughput tissue processing pipeline that I've been working on here at Active Motif in the lab. Really trying to get the speed up for processing mammalian tissues into usable chromatin samples that can be used for ChIP-seq to try and make epigenetics a little bit more amenable to clinical medicine, translational medicine, and personalized therapeutics. So I'm sure myself and the rest of Active Motif, and hopefully everyone watching out there, is a really big epigenetics enthusiast. And I'm sure that we're all interested in epigenetics from a basic science biochemical perspective, but also I'm sure we can all appreciate that there is tremendous potential for epigenetics in being toggleable in the clinical setting. So this was a image that I found from a recent review in 2017 showing how epigenetics could be targeted in cardiovascular diseases. But a lot of these principles hold true for really any disease state. So we can see here from this image that we can target DNA methylation with DNMT inhibitors. We can target histone modifications with HDAC or histone deacetylases or histone acetyltransferase inhibitors, which have broad effects on gene transcription. So in this way, we're really able to target what gene expression looks like in these disease state cells by targeting their epigenetic regulators. And in this way, we can have a much wider impact on what the transcriptional network is doing in these disease states and hopefully lead to more targeted and improved therapeutic outcome. However, despite all of these possibilities, clinical implementation of epigenetic data has been really slow. And I'm sure a lot of us have been on the ENCODE website and have looked at epigenetic data that gets published often in high impact journals and seen that there's a wide availability of epigenomics data, but personalized medical therapeutics have been really slow to implement in the clinics targeting epigenetic regulators. And well, why is that? First of all, there's sometimes very limited clinical samples. A lot of these disease states, you need to get large numbers to make conclusions on a population level, and you can only get so much tissue or material from vulnerable populations. And as a result, there's often not enough material to answer relevant questions. Oftentimes, you only get a couple mgs or maybe a gram of tissue. You process that into a chromatin sample, and you realize upon your input prep that you're not going to have enough to do a ChIP-seq experiment. And in that case, you can't actually ask any questions that are going to be relevant to figuring out what might be targetable in these disease states. It's also extremely difficult to make chromatin. I'm sure a lot of people out there would agree that a chromatin prep is cumbersome and can take a lot of time. And as a result, you would need an army of epigenetic scientists constantly making chromatin from these samples. And often these chromatin samples then need to be sonicated for ChIP. And if you have inconsistent shearing of your samples, you could end up having to throw away entire chromatin preps just because they're not the right size. And I know that inconsistency in shearing has been a major issue for epigenetic scientists. So we need to find a way to standardize the shearing and know that each time we sonicate a chromatin sample, we get high quality, good DNA of the right size that is amenable for ChIP. And lastly, I wanted to point out that there are oftentimes barriers in tissue homogenization. We are intact organisms, and our tissues do not want to become single-cell suspensions. And for the most part, that's a really good thing because it keeps us alive. But from a chromatin material perspective, this can be a major issue because if we can't make a single-cell suspension of the tissue, we have a low yield of cells, there's low amounts of DNA, and we circle back on the first point where now there's not enough material to answer the relevant questions. So all of these issues have become major problems in terms of implementing epigenetics into the clinical space. And part of this project is hoping to standardize a pipeline that makes clinical tissue processing faster, more standardized, and consistently reliable so that we can ask high impact epigenetic questions and understand how we can more accurately target these disease states. So how do we bring epigenetics from the bench to the bedside? That's the major focus here. And we need a high throughput, consistent method for processing mammalian tissues for chromatin for epigenetic studies. We need to know that we can do it quickly, the first time, and correctly every time, so that we can make the most of these precious samples. So one of the techniques that I've been playing with here in the lab is called a rapid bead-based homogenization. And for this, I've been using a machine called the Omni Beadruptor, which is a high-speed bead-based homogenization machine capable of generating single-cell suspensions from a number of different mammalian tissues. And for these initial experiments, I did this in mouse tissues. So I tested the capabilities of 3 different homogenization buffers to homogenize this tissue. 2 different bead sizes. I used a 2.4 millimeter and a 5 millimeter bead, which you'll see the data for in just a little bit. And I did this in 7 mouse tissues. I did this in spleen, kidney, liver, brain, heart, lung, and colon. And for the sake of brevity today, I'm really just going to be zooming in on the data that's been generated from mouse spleen. So first, I just want to show everyone that the bead beating can yield a homogeneous single-cell suspension, and this is mouse spleen. So what we're looking at here is this is a bright field image of trypan-stained single cells generated from bead homogenizing mouse spleen. And I know it can look a little weird. There's a lot of gray dots, so I just kind of want to draw a circle around the region. All of the gray dots in that region are either single cells or intact nuclei. At this point, it is a little bit difficult to tell whether we have a single cell suspension or we have a nuclear isolation because there's no unhomogenized control that I can really put on a hemocytometer. So we have to do some further studies to figure out exactly what these membrane-bound bodies are. But in some data that I'm not showing, you can spin down these samples and do a buffer exchange, and you don't lose any genetic material, implying that this homogenization process does not lyse the cell and release nucleic acid that gets lost in the homogenization buffer. However, one of the buffers that the homogenization takes place in is the same homogenization buffer, which I'll show you in just a second. So in theory, all of these samples can go from homogenization to sonication in the same step, with no spin step required. So now that we had generated these single cell suspensions, I wanted to sonicate the material to generate usable material for a ChIP-seq experiment. So to sonicate the material, I've been using the PIXUL Multi-Sample Sonicator. Our PIXUL Sonicator is a new product here at Active Motif that we have been extremely excited about. And it is a simple, rapid, and extremely consistent method for shearing chromatin, DNA, RNA, and protein. So if we look here, this is the PIXUL instrument. It's actually a really nice size instrument. It fits right on a desktop. It's very user-friendly in that it has a touch screen available and all the parameters are easily set and visible for you. So just to highlight a little bit what the PIXUL does, it is a 96-well plate-based sonicator. So this uses U-bottom 96-well plates to sonicate material. So in theory, you can do 96 samples all in the same run, which is really amenable to clinical translational medicine, as they will need to process a large number of samples simultaneously to generate the material necessary for their experiments. It is a consistent and high throughput method of shearing chromatin. You can do multiple parameters in one run. Each column in the plate is a 96-well plate, so there are 12 columns. Each column is controlled by its own pair of conductors, which means that each column can run on its own protocol. So in this way, you can do chromatin shearing optimization experiments in a high throughput method, as well as sonicate samples that require different sonication conditions all at the same time by just putting them in different columns. It is extremely inexpensive in terms of the consumables that it uses. It uses a basic standard tissue culture 96-well plate, which many labs out there have large boxes of, so it's not expensive to run PIXUL. And I just want to point out one more time that it allows for sonication of clinically relevant numbers of samples at one time. And this is the most exciting part, is that it can sonicate so many different samples under so many different conditions, consistent and reliably, all at one time. And I'll show you some of the data from that in just a second. Before we go in, I thought it'd be a good time to talk about what sonication actually is. So sonication basically takes place when we pulse these compression waves through a sample. And these waves will cause bubbles to form. that will expand over time. And as they reach a terminal size, they will implode, and that releases a large amount of heat as well as mechanical force, which then goes on to shear and break up the nucleic acid. So this is really what takes place when we sonicate samples. So now just to show some of the results from what happens when I sonicated that single cell suspension generated in mouse spleen. And what we're looking at here is this is tape station data of the resulted DNA that was purified from the input chromatin. And we're looking at 3 different homogenization buffers, buffer 1, buffer 2, and buffer 3. This buffer 2 is the same buffer that is used on PIXUL. So these samples are immediately ready for sonication after homogenization. Then you can see that I used a 2.4 and a 5 millimeter bead. And when we look at this row here for PIXUL, this indicates whether the sample underwent sonication or not. So N for no PIXUL sonication, Y for yes sonicated. And what I hope you can appreciate is that in all of the unsonicated samples, we don't see a migration of small molecular weight DNA, like around 300 base pairs, like we do in the sonicated samples, indicating that the PIXUL sonication is specific for shearing that DNA. And we can see that in all conditions tested, we generated chip DNA of about 300 base pairs, which is ideal for proceeding with downstream chip applications. So this was super exciting. All of the samples were homogenized at the same time on the Omni Bead-Ruptor in 1 mil of homogenization buffer in these 2 mil tubes that are provided by Omni at 4 meters per second for 20 seconds. So just to backpedal for a second, all of this material was homogenized in 20 seconds, which is extremely high throughput, and basically means that multiple samples of mammalian tissues can be processed inside of 20 seconds and then sonicated on PIXUL, meaning that inside the same afternoon, you can generate high quality chromatin from a number of different mammalian tissues all at one time. If we look at the input DNA concentrations from these preps, we're able to see that just about every condition generates high concentration DNA of about 100 nanograms per microliter. Some do better and some do a little bit worse. This data will need some replicates to understand exactly what the range is in there. But in all conditions tested, there's enough to do at least a 5 or 10 microgram chip-seq reaction in these samples, which is ample material for assaying where transcription factors and histone modifications are within the genome. If we look at how pure this DNA is, much of it is very, very, very pure and very clean. We can see that there are 260-280 ratios of about 1.8, which is ideal for downstream next-generation sequencing applications. And if we quantify the percent of material that's actually sonicated, what I hope you can appreciate is that about 95% of the material is fully sonicated. And I'm drawing attention to the 70% mark right here, as this is what we use internally as a QC metric for good chromatin. If you're going to use your chromatin for a chip-seq reaction, we want it to be greater than 70% sonicated. And in all conditions tested, all sonicated material is greater than 70%, implying that this is very, very high quality chromatin. So having been able to generate such high quality chromatin, or what appeared to be so, I performed H3K9 acetyl chip-seq. So this experiment was conducted using our H3K9 acetyl Abflex antibody, which is a wonderful antibody that does really robust chip-seq. And all I'm going to show here so far is what the NGS library preps look like from these reactions. So if we take a look here at the tape station data of what these H3K9 acetyl chip-seq NGS libraries look like, we can see that this is, by the way, all from the 5 millimeter bead homogenized sample. We can see that in all 3 homogenization buffers tested, the input material generates a nice chip-seq library. It generates a nice input library at about 450 to 500 base pairs, which is what we expect for the kit that we use to generate these libraries. And the H3K9 acetyl chip reactions also generated very nice NGS libraries, averaging at about 450 to 500 base pairs, implying that DNA was isolated during this chip reaction, and it was readily converted into a next-generation sequencing library. Because I had mentioned at the beginning of this webinar that I had done this in so many different tissues, I wanted to show that the chip-seq libraries generated in these tissues are all consistent. So we're looking at ChIP-seq libraries from H3K9 acetyl chip in kidney, spleen, liver, and brain. And we can see that in all three homogenization buffers, we get these really nice ChIP-seq libraries averaging between 400 and 500 base pairs. And the same is also true for ChIP-seq that was done in heart, lung, and colon. So at this point, this process, 4 meters per second for 20 seconds homogenization with a 5 millimeter bead and subsequent PIXUL sonication represents a new high throughput method of homogenizing mammalian tissues that leads to high quality chromatin that can be used in ChIP-seq applications. These libraries are currently being sequenced, so we're very excited to see what data comes out of this, and then hopefully apply this process to human tissues so that we can really start to answer some clinically relevant and interesting questions and develop new targeted personalized therapeutics for in the epigenetic space, or help labs do that. So just to conclude what I've shown so far, bead rupture homogenization represents a high throughput way to generate single cell suspensions of tissues. As I said before, all of the single cell suspensions were generated in 20 seconds, and all of the samples were sonicated on the same plate in PIXUL, meaning that much of this work can take place inside of an afternoon. I personally have processed up to 3 or 4 tissues inside of the same day, which is very, very quick. PIXUL sonication of resulting single cell suspensions generates chip-ready, high-yield chromatin of consistent size. And I think that's the most important part. We have the capacity now to take these precious tissues and ensure that every time we do a chromatin prep, we get a high yield and a high quality that is worth answering interesting questions in the epigenetic space. And that PIXUL sonicated tissue chromatin is chip compatible and generates high quality NGS libraries. We still need to see how these libraries sequence, but I'm very optimistic that we're going to uncover some nice data and expand this pipeline to human clinical samples. So with that, I just want to thank everyone for their attention. I really appreciate you tuning into this webinar and giving me a chance to talk about the exciting work that we have going on here at Active Motif. A number of interesting products are coming out to support the PIXUL Multi-Well Sonicator, so please take a look on our website if you are interested in any high throughput epigenetic kits that are PIXUL compatible. Please take a look at www.activemotif.com and feel free to contact me at tech_service@activemotif.com. I'm always excited to answer questions about any of the interesting science that's going on here in the lab, as well as just in the general epigenetic space. So please don't hesitate to reach out if you have any questions. And thank you so much for your time today.